Good to see everybody. Let me remind you today in laboratory to bring your meiosis lecture outline, because I'm going to do that entire outline as part of the pre-lab lecture today. And there's a handout for the laboratory on as well. It's a laboratory on chromosomes. So of course bring that handout also. Alrighty. Let's see. We were still on mitosis here in the lecture. I was discussing these things called checkpoints in the cell cycle. Let's go over those. And you might remember last time I had the wrong overheads printed here, and I hopefully got the right set today. We shall see. All right, so there's this familiar, hopefully familiar by now, diagram of the cell cycle. The purple parts being the interphase when the cell is not dividing but is preparing to divide, and the white parts being where the cell is actually dividing. Prophase, metaphase, anaphase, and telophase are the parts of mitosis where the nucleus divides, and the cytokinesis where the rest of the cell divides. And what I mentioned last time is even cells that are actively, or I shouldn't say that, maybe I should say it this way, that within the cell cycle, sometimes the cell needs to stop. Maybe it's a type of cell that's just not supposed to be dividing in the body, because not all your cells are actively dividing. Or sometimes maybe the cell is supposed to be dividing, but the conditions are not right. Maybe it's not internally prepared to divide yet. Anyway, so there are these built-in stopping points along the cell cycle where it could stop if it has to. And these are called the checkpoints, and there are 3 of them. There's one at the end of G1 that stops the cell from entering S phase. There's one at the end of G2 that stops the cell from entering prophase, and one at the end of metaphase that stops the cell from progressing into anaphase. And what really stops the cell is simply that the enzymes that carry out the next phase are not activated. And as an example of that, in the S phase, that's where the chromosomes duplicate. And so it requires enzymes like DNA polymerase 3 and ligase and primase and a bunch of other enzymes. And so the cell stops here simply because those ones aren't activated. It can't go into S phase if those enzymes are not active. And likewise, the same thing here. What prevents the cell, what this checkpoint really means is simply that the enzymes of prophase, the enzymes that condense the chromosomes and cause the nucleus to go away and cause the spindle apparatus to form, those enzymes are just not active, and so the cell can't be in prophase if those enzymes aren't active. And likewise, the enzymes of anaphase just aren't active, and so that's what this checkpoint is all about. The cell stops there because those anaphase enzymes are not active. And then what I guess I did last time is then now look at these 3 checkpoints in a little bit more detail. Of the 3 checkpoints, the G1 is the most important, because for the cells in your body that are not dividing, that's where they're stopped. This is sort of the normal stopping point for cells in your body that are not supposed to be dividing. It's sometimes called the restriction point of the cell cycle, or it's sometimes called GO, G0 phase. When a cell is stopped at this particular checkpoint, some people say it's left the cell cycle and it's in GO. The cell is still alive. It's still doing respiration, and it's carrying out all its normal cellular functions, but it's just no longer progressing in the cell cycle. It's just stopped before S phase. Well, here's a good question. So are its chromosomes duplicated or unduplicated at this point? If it's in GO. Unduplicated, right, because the chromosomes duplicate in S phase, and it's not in S phase. It's stopped right there just before S phase. So the cell is just sitting there being a very alive, active cell, but it's not dividing. Its chromosomes are not duplicated. Now, some cells in your body are not in GO. They're just constantly going through the cell cycle. They are just always and continuously dividing. And I gave you an example of that. Skin cells are a good example. Just under the surface of your skin, like you see right here, are a layer of cells that are constantly dividing. They're making new skin cells that slowly get pushed up towards the surface, and eventually those new skin cells eventually flake off. You're constantly losing skin cells off the surface, and so they're being replaced by that layer. But most cells in your body are not constantly dividing, so they are in GO. Some of those cells that are in GO are permanently stuck in GO, and they can never get back into the cell cycle. They can never divide. Nerve cells are a great example of that, and muscle cells also. But many cell types that are in GO can get back into the cell cycle. They can re-enter the cell cycle. And there's a classic example of that is a type of cell called fibroblasts, which you see here. And these are the ones that form scar tissue. If you get a wound and eventually it gets filled in by scar tissue, it's because these fibroblasts re-entered the cell cycle and filled in the wound. And that's what is the scar is. Louis?
So when you work out and your muscles, it's like getting stronger. How does that happen?
Yeah, you're not getting new muscle cells, you're getting bigger muscle cells. If here's a muscle cell, before you start working out, after a few months or a few years of working out, same muscle cell, but it just gets larger. It stuffs itself with even more protein, so it becomes a bigger, more powerful muscle cell, but you haven't changed the number of muscle cells.
So fat doesn't turn into muscle?
Fat does not turn into muscle. Two different cell types. You might shrink your fat cells and increase your muscle cells so your body looks more big and defined, but the number of cells has not changed.
Once your muscle cells have expanded and are grown that large, and then you cease to stop working out, they'll shrink again.
Yes. Go ahead.
But will they be like fatigued, I guess, from that? Like, will they have stretch so they'll be smaller, but like kind of?
That's a good question. I assume somehow the cell is able to shrink its membrane back to the smaller size. Like, it might be like a balloon. You can blow it up to a larger size and let the air back out, and it doesn't really hurt the membrane of the balloon. But I'm not 100% sure. It's a good question.
So can a muscle cell grow like however big it wants? Or is there like a stopping point that it'll just
I'm sure there's an upper limit. I mean, if nothing else, it eventually gets so crowded with the other muscle cells it may stop growing. I guess I'm not 100% sure on that question either. Talk to a professional physiologist. Anyway, so let's focus in on cells like fibroblast cells that are usually parked in GO but can re-enter the cell cycle. For this type of cell, for the ones that can get back into the cell cycle, what they have is a receptor on their surface that can bind to, that can detect molecules called growth factors. Growth factors are just signal molecules that tell the cell in GO that it's time to move back into the cell cycle. In other words, they are signal molecules that promote mitosis. And there's hundreds of different growth factors that have been identified because they're specific for different types of cells. In other words, a growth factor that stimulates fibroblast cells is not the same growth factor that might stimulate white blood cells to divide. And the growth factors that stimulate white blood cells to divide are not necessarily the same growth factors that will cause other cells to divide. You know, each growth factor is specific to a specific receptor on a specific cell type in the body. All right, so when the growth factor binds to the receptor of the cell that's in GO, it causes that cell to go back into the cell cycle, which in this case is going to mean it's going to activate the enzymes of S phase, so that now the cell is going to start duplicating its chromosomes and then proceed to G2, and then if everything works right, proceed through the rest of the cell cycle. Like that. Matter of fact, you can see a little cartoon there. When the cell has gone into S phase, DNA polymerase 3 and all those other enzymes get activated, and so it goes through S phase. That's what the GO stopping point is all about. Let's look at the checkpoint at the end of G2. We'll skip this for now. We'll come back to that blob of text here in a few minutes. The G2 checkpoint halts the cell cycle. It's kind of it's a DNA checkpoint. The cell halts there to make sure that the DNA is prepared to go into mitosis, prophase of mitosis. One thing that it checks for is to see if the DNA is duplicated, if the chromosomes have been duplicated, and if usually they have, because that's what S phase is all about. But anyway, this part of this checkpoint is to make sure that the chromosomes are duplicated. Another purpose of this checkpoint is to make sure that the DNA has not been damaged, because some things like getting sunburn, for example, there we go, do damage your DNA. And the cell has enzymes that can repair DNA damage, but those enzymes take time to operate. And so the G2 checkpoint will halt the cell cycle until the mutations, until the damage has been repaired. All right, and if the chromosomes are duplicated and the DNA damage is repaired, then the cell moves past the G2 checkpoint and enters prophase and then metaphase. The checkpoint at the end of metaphase verifies that the spindle is properly attached to all the chromosomes. Remember, the spindle fibers are these microtubules here and here, and each chromosome has to be attached to spindle fibers that attach it to the bottom centrosome and also to the top centrosome, because here in a moment in anaphase the centromeres are going to split in two and each chromatid will move in opposite directions. So anyway, the cell is checking here to make sure that each chromosome is attached to the top pole and the bottom pole of the cell. And if that checks out, then the cell gets to move past the metaphase checkpoint and then proceed through anaphase and telophase and cytokinesis. And when the cell has gone through a round of the cell cycle, all the checkpoints reappear. So in other words, think of it this way: the cells are automatically designed to re-halt after going through mitosis, unless the signal molecule is still present. In other words, the body very tightly regulates mitosis, and only if the signal molecule continues to be present, only then is the cell allowed to stay within mitosis and keep going around and around and around. Okay, and one more little thing about this, the checkpoints. So when I was telling you about this checkpoint here, the G1 checkpoint, I said that's the normal stopping point for cells in the cell cycle. That's where they're supposed to park themselves, so to speak. Maybe a good analogy is like the parking structure outside is where you're supposed to, it's the normal parking spot for cars around here. The G2 checkpoint and the metaphase checkpoint are not normal stopping points, because they only stop there if something's wrong. Only if the DNA is damaged or the chromosomes are not successfully duplicated do you get any stopping here, but that doesn't normally occur. And likewise, only if the microtubules of the spindle apparatus have not been attached to the chromosomes do you get any stopping here, but normally the spindles do attach successfully, and so that's not a normal stopping point. So think of these as like emergency roadside stopping areas along the highway. You're not supposed to stop there normally, only under very unusual circumstances. But this is the kind of standard normal stopping point. Okay, in fact, because the GO checkpoint is so important, I want to look at that one in a little bit more molecular detail and see how it operates. So there's the cell stopped in GO, and let's now give it its growth factor molecule. Now let's kind of have a close-up of that cell. There in pink in the upper left, you see the receptor for the growth factor about to bind the growth factor. And here's a little mini diagram so we can see where we are in the cell cycle. We're halted in GO. And this enzyme right here is going to represent the S phase enzymes, the enzymes that that carry out S phase, that like DNA polymerase 3 and ligase and primase and all the other enzymes. Notice that it's black to indicate it's in an inactive state, because that's what I was telling you, that when the cells are stopped here, they are stopped here because these S phase enzymes are not activated yet. And so the goal for the cell is that when the growth factor binds to this receptor here, what we're looking for is eventually to activate the S phase enzymes so that the cell does enter S phase. Well, between the receptor and the S phase enzymes are these guys right here, and those should look familiar. They are signal transduction pathway proteins. They are relay proteins that are going to carry the activation, so to speak, from the growth factor binding the receptor to eventually leading to the activation of the S phase enzymes. And if you remember back to the signal transduction chapter, most of these relay proteins between the receptor and the cellular response enzymes are kinases, that each one activates the next one in the pathway by adding phosphates to it, and that activates the next protein. So let's watch that in operation. So when I click here, the growth factor is going to bind to the receptor, and this first protein in this pathway is going to get activated right here. And